there's been a huge rise, in studies of the human brain. And of course, that's due to the development of brain imaging. And it started in 1990 when the bold signal was discovered, and that made FMRI possible. There are other ways of using FMRI, but that's the standard one. But the question is whether cognitive neuroscience, which developed as a result of brain imaging, has fulfilled its promise. Now, to answer the question, let's compare it with other imaging methods. Take the electron microscope. 20 years after the electron microscope was first developed, it was able to show that there are packets of neurotransmitter in the presynaptic site, and you can see them there, and also you can see the synaptic cleft. And that proved what had often been thought but never proved, that transmitters pass through the synaptic cleft to cause postsynaptic potentials. Or take the radio telescope. Now, this is the one that Jocelyn Bell built in Cambridge. And what she discovered was repeating radio waves coming from space. And they turned out to be pulsars, which are rotating neutron stars. And this was a totally new discovery. So the question is, has FMRI discovered anything that's similar, that's fundamental about the brain? Well, what do I mean by fundamental? I'll give you 3 examples. The first is that Hubel and Wiesel showed that simple cells in the visual cortex feed onto complex cells, and that complex cells then fire in response to lines of specific orientations. And Bob Desimone then showed that throughout the whole of the ventral visual system, there was a similar hierarchical arrangement. And that led Poggio to suggest that this would enable object recognition. So if you look at the figure on the right, at the bottom is the input shape, and then cells in V1 responding to lines of particular orientations. But by the time you've gone through the hierarchy of the ventral visual system, and you're in IT or infratemporal cortex, you find cells that respond to shapes. And those have been discovered by Tanaka and his group. My second example of a fundamental discovery is the distinction that Leslie Ungerleider and Mort Mishkin made between the dorsal and ventral visual systems. The suggestion was that the ventral visual system is specialized for identifying objects and the dorsal for locating them. And part of the evidence was that the ventral visual system gets a foveal representation, whereas the dorsal gets a peripheral one. And if you locate an object, whether it's yourself or another object, you locate it with respect to the periphery. And of course, Milner and Goodale suggested an alternative possibility, which was that though the ventral visual system is specialized for object recognition. The dorsal is specialized for using vision for action. And the evidence for that was that the dorsal visual system projects to premotor cortex. And the third fundamental discovery, as an example, is that made by Larry Weisskranz, and that's blind sight, which is the dissociation between awareness and performance. And it was first discovered by studying a patient called DF, who had a lesion in his left primary visual cortex. That means that he was blind for anything shown to the right, in other words, blind in his right hemifield. Now, the task was very simple, and it was simply to point to an object. This shows the position of the finger in degrees and the position of the target in degrees. And if you were pointing accurately, then the data would lie on a line at 45 degrees. And the solid line shows you the data when the stimulus is presented in the intact field. And as you can see, astonishingly, you get the same result when it's in the blind field, even though patient DF saw nothing in the blind field. And this finding was of enormous impact because it led to the scientific study of consciousness. So the question is, has brain imaging really led to comparable results? Well, in 2012, Stephen Wise and I wrote a book called The Neurobiology of Prefrontal Cortex. And in this, of course, we discussed the organization and functions of prefrontal cortex. And all but one chapter were based on evidence from macaque monkeys. We found absolutely nothing from FMRI studies of the human brain that changed our view of prefrontal cortex. There was a chapter in the book on it, but all it did was basically say that the human brain was organized in the same way as the monkey brain. Now, I'm not denying that brain imaging has done impressive things. After all, the image consists of voxels. And as you can see, the activations go across voxels, and you can see that by the yellow circle. And that means that you can analyze the pattern of activation across voxels. And you can do that in one of two ways, by using multivariate pattern analysis or by using representational similarity analysis. And you can do very impressive things that way. This is data from Haxby and Connolly, which shows that you can classify from looking at activations in the brain what the person is actually looking at. Now, the stimuli are shown at the left in a vertical line, and the classification is shown along the bottom. So, if you look at the bottom far right, that's a house, and the color scale tells you how well it's classified. And the answer is 90%. The next one is a shoe, and that's yellow, and that means between 60 and 70%. A chair, 90%. and so on. In other words, very impressive ability to classify what you're viewing. So there's no doubt that you can do clever things. The only thing is that this had already been shown by optical imaging in macaque monkeys, where at the top you see the maps for different stimuli shown as these circles, and the stimuli are shown at the bottom. So, what are the advantages of fMRI? And in particular, do these mean that it can discover new principles of brain function, i.e., change the way we think about the brain? Well, the first advantage, and I'm going to suggest two, is of course that it shows activations over the whole brain. It's a whole brain method. That means that you can measure the covariance in activation between areas, and the arrow in blue shows you co-activation between the superior temporal cortex and Broca's area, and they co-vary because they're connected via the arcuate fasciculus, and that's shown here by using diffusion-weighted imaging to look at the white matter. So imaging can visualize networks. What does that enable us to do? Well, one of them is the so-called default network, so-called by Marcus Raichle. And this shows the activity on the medial surface for the default network. Now, it's defined as the areas that show a decrease in activation when the person does an externally specified task. The same areas show an increase when you do an internally generated task, as in mind wandering. So there is a principle here, and the principle is that there are decreases in areas that are irrelevant for the current task. And the data here come from Ramnani and myself, and it's a motor learning task. You're pressing a finger in a particular rhythm, and it's auditorily paced. And of course, vision is irrelevant for that. And we measured blood flow using PET, and there are 6 stages of learning, as you can see. And as you can see, as learning progressed, there were further decreases in blood flow in the infratemporal cortex. It's irrelevant for the task. The next principle is context-specific connectivity in networks. This was first suggested by Randy McIntosh using structural equation modeling, and has then been further developed by Carl Friston using dynamic causal modeling. I'll give you an example, and it comes from work that Katsaka and I did a long time ago. The task is that you're going to be presented with a word. But before you're presented with a word, you're presented with an instruction which tells you which task you're to perform on the word. One is that you're to say whether it's 2 syllables or not. And the other is to say whether it's abstract or not. So if the task was 2 syllables, the answer is yes. And if the task was abstract, the answer is no. And we measured activation in FMRI during the delay period after the instruction and before you get the word. And this is what we found. During the delay, if you measure the correlation R between activation in the polar prefrontal cortex, you find greater activation between polar prefrontal and premotor than Broca's if what you're going to do is the syllable task. And the reason is that ventral premotor cortex is involved because you are silently sounding out the word to find out how many syllables it is. On the other hand, if you're doing the semantic task, then polar prefrontal now correlates higher with Broca's area than with premotor cortex. And that is because you're doing a semantic analysis. So the principle is that the connectivity of areas depends on the task, here shown for the syllable task. Now, the delay activation, we thought, reflected task-dependent preparatory set. That's what it appears to do. It appears to be setting up the task. But the data depended on correlations. And that's true of all the data from FMRI. And I put a ring round R just to strengthen that point. So then the question comes up, is the effect causal? And to find that out, you need to intervene. And one method of doing so is to use magnetic brain stimulation, or TMS. So in a study by Heinen et al., done with John Driver, the task was as follows. You were going to see a face, and it was made up of dots that were moving. And the instruction was either that you were going to tell whether the dots were moving in one direction or another, or you were going to say, was it a male or a female face? And the TMS, which I'll show you, was applied during the delay before you were shown the face. So there you are, the TMS pulse was given to the frontal eye fields, and the reason it was presented there was we know the direct projections both to the motion complex, MT+, and to the fusiform face area, shown in red. And the data were as follows. TMS to the FEF enhanced activation in the task-relevant area. So if the instruction was that you were going to do the motion task, TMS enhanced activation in MT+, but not the FFA. On the other hand, if you're going to do the gender task, the face task, TMS enhanced activation in the FFA and not MT+. So indeed, it's causal. Now, the third principle discovered by imaging as a result of being a whole brain method is that there are changes in grey and white matter with experience. And that was first discovered by Eleanor Maguire in a study of London taxi drivers, where, as you can see here, she showed that there was an increase in grey matter in the posterior hippocampus as a function of the time the taxi driver had driven, measured in months from 0 to 400. And this is so surprising that most journals simply rejected it. They didn't believe it. And it only got into PNS because a very famous psychologist believed it. And it turns out that, in fact, she was right. The finding is general. And you can see that by a study done in Heidi Johannesson Berg's lab by Schultz et al., in which they got people juggling for 6 weeks. Now, there are 3 training sessions, 1, 2, 3, which you can see along the bottom. Now, you don't know where and if there are going to be any changes, so you need a whole brain method. The measure taken is fractional anisotropy, which is a measure of the degree to which water is passing in the same direction as opposed to all directions. Is it flowing, in other words, along axons? And there was an increase in this measure, Fi, in the parietal white matter. And in studies that they carried out later with Dave Manneman in the department on rats, they showed that this was due to an increase in the amount of myelin. So the principle is that there are changes in conduction speed with learning. Now, there's a last principle that I want to illustrate, and I want to illustrate that by comparing the human and macaque monkey brains, and using the fact that FMRI tells us where the activations are. And I'm going to specifically compare the dorsal with the ventral prefrontal cortex, and then at the end, I'll tell you what the principle is. So if you want to look for activation in the dorsal prefrontal cortex of the human brain, one way of doing it is to use the Raven's matrices, an IQ test. And what the people do is they look at the first line, it's a series, and then they look at the second line to try and see if the same series obtains. Now, of course, the material in the second line is different, so it's an abstract series. And then their job is to fill in the last item in the bottom line according to the series. Now, you get the same data if you use letter series or number series, but I'm illustrating it with these particular sequences. And what you find is activation in the dorsal prefrontal cortex area 946 and activation in area 46, which lies more anteriorly. Now look at the macaque dorsal prefrontal cortex. And this is an experiment by Tanji's group in which the monkeys had 3 movements to make: to pull, push, or turn a handle. And along the bottom, you can see they're to make 4 movements, in this case, turn, turn, pull, pull. And what they found were cells that coded for abstract sequences. So I need to explain that. The cells in green here code for alternating movements, irrespective of what the actual movements are. So now if you look towards the right, you can see the same cells code for turn, push, turn, push, turn, pull, push, turn, and pull, turn. And they're found in the dorsal prefrontal cortex. Now look at the ventral prefrontal cortex for the human brain. And one way of showing activations is to test for associative knowledge or semantic knowledge. And this test, given by Kathy Price, is taken from the pyramid and palm trees test. And the question is, which of the two bottom objects is associated with the top one? And the answer is the spanner, because the spanner, like the pliers, can be used to turn things. And when you give that, and of course there are many items on the test, you get activation, as pointed out here, in the ventral prefrontal cortex as well as the infratemporal cortex. Now look at the macaque-monkey ventral prefrontal cortex and a test for associating pictures. So here the monkey fixates, picture 1 is shown, there's a 1 second delay, and then picture 2 is shown. And the monkey's job is to learn, given picture 1, what's coming up is picture 2. And here is the data. You find activity in the ventral prefrontal cortex for associating pictures. And this shows a cell with prospective activity for picture B, given picture A. Now the black band shows you the time at which picture A is shown, and the ascending activity shows you the prospective activity as the monkey expects picture B. So the monkey has successfully learned the association. So in sum, the dorsal prefrontal cortex codes for abstract sequences where the order is critical, and the ventral prefrontal cortex for associations irrespective of order, e.g. whether A goes with B or B goes with A. And the principle here is that our brains have co-opted mechanisms from the common ancestor that we share with macaque monkeys in this case. And that explains why the activations are where they are. Now, I said there were 2 advantages of imaging, and the second one is plainly obvious, and it is that we can image the human brain. We can study the human brain at work. And what that means is that we can study abilities that are unique to us. I'll give you some examples. Episodic memory. When we retrieve memories from the past, you find activations on the medial surface in the areas shown. And the same areas are activated when instead what you do is imagine events in the future. And Schachter has called that episodic simulation. And the idea is this: when we retrieve a memory from the past, we re-experience it in just the way we do if we imagine or experience an event in the future. Now, what is special here is that we can think about events in the distant past, or indeed distant future, for example, 10 years ago. And this data by Bonici and Maguire shows you data in which they classified what memories people were thinking of, and the accuracy is shown on the ordinate. And the memories were either laid down 2 weeks ago or were from 10 years ago. Now, along the bottom, I've labeled the different areas and we'll only look at 2 of them. At the far left is the hippocampus, and you can see that the accuracy is very similar whether you're remembering from 10 years ago or 2 weeks ago. Now look at the far right, and that's the prelimbic cortex. And now you're much better at classifying memories for 10 years ago as if they're laid down or consolidated there. Next, theory of mind. Now, this shows activations when we're thinking about our own mind or the mind of others. And that's shown in yellow on the medial surface here. It's not clear whether we can think about the minds of others because we can think about our own minds, or, as some people think, the opposite is true. But what is special is that we can explicitly represent mental states, not implicitly. We can explicitly do so. Then, of course, we can reason. This shows activation when you're reasoning about syllogisms, of which an example is shown at the top. All men are mortal. Socrates is a man, so Socrates is mortal. And the question is that true? That's what the person has to judge. And in the control for this, reading is taken out, is subtracted. And that's done by having all men are mortal, Socrates is a man, but then having a third line which simply has nothing to do with it. And when you do that subtraction, you find that when you do the syllogism, there's activation in Broca's area, and there's activation in the superior and middle temporal cortex. Now, very importantly, the same areas are activated during inner speech. By inner speech, what we mean is that we're thinking to ourselves silently. Now, we're not thinking in sentences, but nonetheless, we're thinking in words. And what you can see is that when we do that, there's activation in Broca's area and the middle temporal, just as there is when we're doing the syllogism. So what is special is that we can reason using what Coslin called a propositional code, not just a visual one, as a monkey would be able to do. And by a propositional code, he did not mean sentences. They're not sort of fully flown sentences, but nonetheless, they form propositions. Finally, there's the case of precise imitation. We can imitate precisely. Now, this is a meta-analysis done by Kaspers and his group of imitating hand movements or arm movements. And as you can see, in green, there's activation in Broca's area. And just to prove to you that there really is, here in another study, is activation in Broca's area when you're imitating hand movements in red or speaking in green or imitating speech. Forgive the growly voice, I've got a cold. So what is special is that we need to copy signs specifically if we're to learn sign language, and we need to copy speech precisely if we're to learn spoken language. Well, of course, given that we can study the human brain, we can also study disorders that are unique to us. One is a speech problem discovered by Farineh Varga Khadem and Kate Watkins in members of the K family in London, a 4-generational family, and their speech is severely impaired. And the data shown at the bottom shows areas that are underactivated when the affected members of the family generate verbs that are appropriate for nouns. Broca's area, the putamen, parietal cortex, and premotor cortex. In schizophrenia, people sometimes hear voices, and when they do so, as you can see by the circled activity, there's an activation in auditory areas in the superior temporal cortex. And my third example is depression. And what's critical about depression in the human brain is that because we're able to think about the past and the future and the distant future, we're able to brood. And brooding makes depression very much worse and long-lasting. And what Wayne Drevitz showed using PET in 2000 was that there was underactivation in the subgenual cortex in people who were depressed. But, and it's a big but, what we want to know is how the brain works. So labeling and locating is really just the first step. It doesn't tell you how the mechanism works. And I'm illustrating that here with a car engine. It's beautifully labeled and located. So there's a fan belt and there are spark plugs. But you can know all of that without knowing how it works, how the pistons are fired and so on. So all we've done so far is really just labeled and located. Yet what we need to know is how the brain works, what's its mechanism. And so there's a moral here. You really must not see yourself as an FMRI scientist. You're a scientist, and your job is to solve problems. And you must use all the techniques needed to solve the problem. FMRI, EEG, and MEG, which have millisecond resolution in time. TMS or RTMS. RTMS can be given at theta frequency, bilaterally, to knock out temporarily while you do it before you give the task, and then while the people do the task, there's still underactivation in the areas that have been knocked out. Then there's MRI spectroscopy, in which you can measure things like glutamate and transmitters. And of course, there's cell recording directly in patients, which is increasingly happening because surgeons performing surgery on patients with epilepsy often implant electrodes in the weeks before surgery. And we now have data across most areas of the brain now, if you go across studies, for cell activity. So how do you solve a problem? And I'm going to take as an example, how do you perform a voluntary action? And the problem was set by John Searle, the philosopher, who's raising his arm voluntarily and points out that there's a difference between raising your arm and your arm rising. And the problem matters because Parkinson's patients may walk slowly, but then sometimes they simply freeze. They're unable to walk even though they want to. So a long time ago, we taught monkeys to do John Searle's task. They raised their arm in the dark. That meant that the movements were self-initiated because there were no cues. The food arrived in a food well below, so they're not reaching up for the food. It's simply that when they reach up, the food arrives below. And they do that whenever they want, and the data look like this. These are data for monkeys that had lesions of the SMA and pre-SMA. First of all, the data before surgery are shown by the clear bars, and you can see for 3 monkeys that they raised their arm roughly 10 or 12 times a minute. But look after surgery, almost not at all. And it's not because they couldn't raise their arm, because if we put a peanut up at the top, which they could see, they reached for it. And we got the same results with lesions of the cingulate sulcus. But where is the activation for the decision? And to find out, Hakwon Lau and I gave the Libet task. You press a button whenever you want, and you have to remember the time on the clock shown at the left when you made the decision. That blue spot is going round the clock very fast. And then 4 to 10 seconds later, you move the central spot to the point at which it was on the clock, as you remember it. And we compared timing when you made the decision or timing when you made the movement, and the difference for the decision was activation as shown here by the activation in the circle in the pre-SMA as it borders the cingulate sulcus. But is the enhancement for the decision constant, or does it increase? We'll need to use EEG. And Rigoni did that. And in black, you see what happens before the decision when you're timing the decision, and in the light color when you're timing the movement. And you can see that there's clear increasing activity, the readiness potential, which comes from the SMA, pre-SMA, and the underlying cingulate sulcus. But does the cell activity align with the decision? Well, you need to record from cells in patients. I've put a big bar for the decision in blue, and you see spikes per second from 2 seconds beforehand up to the decision. And these are taken in the SMA, pre-SMA, and cingulate sulcus by Fried. And as you can see, there's activity that increases up to the point of the decision. But is the key area the pre-SMA or the cingulate sulcus? Well, you can scan monkeys with FMRI. And that was done by NEMA. And as the bubbles go down, the monkeys decide when to press the key. It's their decision. And the activation for deciding to act was in the anterior cingulate sulcus. As you can see, it's not the only one, but that's the one that I'm pointing out here. And it's very rostral, i.e. very anterior. But why should the anterior sulcus, cingulate sulcus, be a key area? Well, to find that out, you need to record from cells in the area, as Lawrence Hunt did. And what he found was cells that fired both for the reward and for commitment to action. And if you think about a self-initiated task, like the arm raise, is anticipation of the reward that leads to the self-initiated action. So the cingulate sulcus at this area has exactly the properties needed for self-initiated action. That doesn't prove that the cell activity is causal. To find out, you need to inactivate that activity, which they did, using focused deep ultrasound focused on the anterior cingulate sulcus. And that influenced the time to act. But of course, it's not the only area, as I've already pointed out, and you can find that out, as they did in a further study done on people using FMRI. It's a whole brain method. And I'm pointing out this, that there's an increasing signal in the substantia nigra preceding the action. You can see that in the plot. And this is a dopamine signal, and it reflects motivation to act. So solving the problem involved behavior, human fMRI, monkey fMRI, EEG, cell recording in monkeys, cell recording in patients, and focused deep ultrasound. So, has brain imaging discovered anything new about the brain? And as I've told you, in 2012, I was not persuaded that it had. But Marvin Minsky said, if I still believe something after 5 years, I doubt it. And I'm beginning to have my doubts. So long, of course, as you use all the techniques. Thank you.